Welcome to AI Explains.
Great to be here.
We're going to talk about the human genome today. So I want you to imagine a massive instruction manual, maybe the biggest one ever written.
Billions of letters long.
Exactly. 3 billion letters. And it's written with only 4 letters, A, C, G, and T. Now, what if a single typo in that book, like an A where a G should be, could cause a serious disease?
Which is a reality for so many genetic conditions. And that brings us to today's deep dive. We're looking at a groundbreaking paper from Nicole Gaudelli, David Liu, and their colleagues.
At Harvard and the Broad Institute, right?
That's the one. They developed something called adenine base editing, or ABE.
And this isn't like the CRISPR you usually hear about. That's often described as molecular scissors. This is different.
Very different. Think of it less like scissors and more like a molecular pencil.
Okay, a pencil. So you can erase an unwanted A and just write in a G without cutting the paper?
That's the perfect analogy. No cutting. That's the key. Because the scissors approach has a big problem.
Let's talk about that. The standard system, Cas9, it literally cuts the DNA double helix. It just snaps it.
Right. And when that happens, the cell panics and tries to patch it up.
And that repair process is, it's messy. The data in this paper really highlights that.
It's incredibly messy. The paper talks about indels. These are insertions and deletions. The cell, in its rush to fix the cut, might add a few random letters or lose some.
So you try to fix one typo and you might just scramble the whole sentence. The source data showed the error rates with the old Cas9 method could be, what, pretty high?
Oh, definitely. Sometimes over 10, even 20%. That's a huge risk. You're trying to perform surgery and you end up causing more damage.
Which is why this pencil idea is so revolutionary. But there was a big hurdle.
A huge one. Nature didn't really give us an enzyme that could do this specific job, turning an A into a G inside a DNA.
It just didn't exist.
Not for DNA, no. But a similar protein called TADA did exist for RNA in bacteria like E. coli.
So the team couldn't find the tool they needed. So they decided to build it. Or, maybe more accurately, they forced it to evolve.
That's the most incredible part of this whole story. They used directed evolution.
How did they even do that?
Well, they set up a brilliant system. They took bacteria and gave them a broken gene for antibiotic resistance. The only way for the bacteria to fix that gene and survive was to change a single A to a G.
It's survival of the fittest on a microscopic level. Evolve this protein or you die.
Exactly. It was a brutal but effective gauntlet. And if you look at the supplementary tables in the paper, you can see the progress. They went through 7 rounds of this.
And that's how we get to the final version, the champion, ABE 7.7.
ABE 7.7. The jump in performance from the earlier versions was it was stark. It was way more efficient and precise.
So they took it from the bacteria and put it into human cells, right? The HEK cells mentioned in the study.
They did. And ABE 7.7 worked beautifully. It successfully flipped that A to a G with really high efficiency, often around 50%.
Which is great. But the real game changer for me was the safety data. What about the indels? The messy edits?
That's the takeaway. They were almost non-existent.
Really?
The data tables show indel frequencies of less than 0.1% in most cases. It fixes the typo without wrecking the sentence.
So we've moved from genetic surgery to, what, genetic chemistry?
I think that's a great way to put it. We're not cutting anymore. We are rewriting letter by letter.
Which opens the door to treating that whole class of diseases caused by these single-letter mutations.
A massive class of diseases, yeah.
Yeah.
The possibilities are enormous.
So to wrap up, here's a thought to leave you with. If we can now safely correct these tiny single-letter typos in our DNA without actually cutting the strand, are we looking at the beginning of the end for genetic diseases caused by these point mutations?
It's a scientifically grounded possibility now, and it's certainly one worth watching.
Thanks for joining our deep dive. Stay curious.